Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting, and I have another great lesson ready for y'all today, okay? We're talking about enzymes, which are a type of protein, and they are very, very important to the functioning of your body. So without any further ado, let's go ahead and hop into it. So first of all, what is an enzyme? Enzymes are biological catalysts. Well, what exactly does that mean? A catalyst is something that's going to change the rate of a chemical reaction without being changed by the reaction itself. What that means is that we can use enzymes over and over again because they're not consumed by the reaction that they took place in. Okay, enzymes are very important because they lower the energy of activation. Another way of saying that is that they lower the activation energy. Well, what does that mean? Well, imagine I asked you to activate your car or your cell phone. What does that mean? To activate something means to start it, right? So the activation energy is the amount of energy it takes to start a chemical reaction. Because enzymes lower the activation energy, it means these chemical reactions can take place with less energy input overall, okay? So, to review that information, enzymes are proteins. That's very important. Let's remember that proteins are the most diverse and complex macromolecule. They have a lot of jobs in the body, and one of those very important jobs is that they are enzymes. What do enzymes do? They speed up chemical reactions, again, without being consumed by the reaction itself or using energy. So we can use these enzymes over and over again. At the bottom here, I have a couple of different examples of enzymes. So you have amylase. This is a type of enzyme. It's in your saliva, and it likes to break down sugar. Specifically, it's going to break down starches into simple sugars. That's why if you're chewing on bread or crackers, it'll kind of start to taste sweeter in your mouth as you're eating it because it's being broken down chemically. Okay, another example of an enzyme is protease. Protease breaks down proteins, okay? The names are kind of similar there. Another example of an enzyme is lipases. Lipases break down lipids. And just like with the proteases and the proteins, lipases and lipids really sound a lot alike, so that's pretty easy to remember. Another enzyme that we're going to be looking at a lot, especially next week, I have a whole lab based on this enzyme. It's called catalase. Catalase is an enzyme in almost all living tissue, and it breaks down hydrogen peroxide into water and oxygen. I wanted to point out something about a couple of these words real quick. So let's look at amylase and catalase. You'll see that they both end with the same three letters, A-S-E. That's a really big clue that whatever you're looking at is an enzyme. You might remember from a previous unit that sugars usually end in OSE, and enzymes, and therefore proteins, often end in ASE. So if you're looking at a word you don't know what it is, if it ends in ASE, it's a good chance that it's an enzyme. All right? So let's talk about the parts of an enzyme and how they come together in a chemical reaction to break something down. So here are a couple of different parts of an enzyme that you need to know about. First of all is something called a substrate. The substrate is the molecule that an enzyme acts on. So an enzyme will come into contact with the substrate, it'll break it down, and whatever that reaction yields, we'll call the product. So the substrate is what you're starting with, and the product is what you're ending with. Okay? Another important part of an enzyme is called the active site. The active site is the spot on the enzyme that the substrate attaches to. I just like to think of it as this is where all the action happens. So action and active, they sound a lot alike. That's kind of easy to remember. So let's take a look at that real quick. Okay, so look at this picture. Down here on the bottom, I've got the blue. That one's labeled as an enzyme. Up top, the one that's purple, I have that labeled as a substrate. So this is an enzyme complex. What happens is the substrate and the enzyme are going to come together. You can see they almost look like puzzle pieces. They fit perfectly together. And then that enzyme is going to break that substrate down. It's going to break it into smaller molecules. And whatever we're ending with is going to be called the product. It is produced by this reaction. So substrates are what you start with, products are what you end with. I did want to take a second and just talk about the shape of this enzyme. I kind of mentioned how the enzyme and the substrate kind of look like puzzle pieces. They fit like perfectly together. So the shape of an enzyme and proteins in general are very, very important to their function. you might be able to tell that if this enzyme wasn't shaped that exact way, that substrate wouldn't really fit into it very well. And if it can't fit into it, it's not going to be able to break it down. So the shape of an enzyme is very important to its function, and that'll come up a little bit later on in this lesson. Okay? So there are two models about enzyme action and how they interact with these substrates. The top one there is called the lock and key model. And the idea here is just like a key fits into a very particular lock, you can't unlock every door with your house key, but you can unlock the lock at your house with it. So just the same way that certain keys only unlock certain doors or certain locks, some enzymes only work on one substrate. And so these are substrates and the enzymes fit perfectly together, okay? So that's called the lock and key model. Another model for this enzyme action is called the induced fit. This is the one at the bottom. You can see that the enzyme and the substrate kind of have similar shapes. They don't fit perfectly together, but in that middle picture, we're inducing the shape. So we're kind of forcing the substrates into the enzyme, and we're still able to break it down that way. So there's kind of two models. This can happen in either way. One is the lock and key, and one is the induced fit. And so just kind of a summary for that. The lock and key model, the substrate and the enzyme fit perfectly together like puzzle pieces, okay? On the induced fit model, the enzyme changes shape slightly to accommodate the substrate. So the shape of the enzyme is very important, and that's the only way it's going to be able to break down the substrate is if the enzyme and the substrate kind of fit together, okay? So I wanted to talk about the role of enzymes in that term I talked about earlier known as the activation energy. Okay? So enzymes are really important because they lower the activation energy needed for a chemical reaction to happen. What is activation energy? Activation energy is the energy needed to start a reaction. To activate something means to start it, and so activation energy is the amount of energy you have to put into the reaction to kind of get it going. Okay? Enzymes are very important. Enzymes are a protein catalyst, and what that means is that it speeds up the rate of these biological reactions by lowering the activation energy. So we're starting and ending with the same substrates and products, but the amount of energy we're putting into this reaction to get it going is a lot less when we use an enzyme. Some life processes are too slow when they occur on their own, but enzymes help to speed them up. If you kind of look at these two pictures here, on the left I've got the pink graph and on the right I got the green graph. Okay, so on the y-axis is the amount of energy needed to make this reaction happen, and on the x-axis we have the substrates and the products. What are we starting with and what are we ending with? You can see that in both of these examples we're starting and ending at the same point. The only difference here, look at those little hills by the balls. Now I don't know about you, but if I was riding my bicycle, I'd rather ride up that green hill because it's not nearly as steep and it would take a lot less energy to climb over, okay? So enzymes lower the activation energy and make chemical reactions happen faster, okay? And again, enzymes are not consumed by the chemical reactions that they make happen, okay? Great job, y'all. So something that's really important, I mentioned this earlier, the shape of an enzyme is critical to its function, but that shape can actually change, and it changes by a couple of different examples. So here are two that I have for you. Temperature and pH, so really, really high temperatures, really cold temperatures, as well as high pHs and low pHs can change the shape of the enzyme or the active site. And what that means is that if the shape of the active site changes, the substrate's not going to fit into it, and that means the enzyme's not going to work. We call that denaturation, okay? You denature the enzyme. When you denature an enzyme, it doesn't work anymore. And this can happen, like these examples show us, in temperature and pH. Now I have a whole lab plan that we're going to look at later on that really drives this point home later in the week. So for now, just remember this term, to denature an enzyme, or denaturation, is when an enzyme doesn't work anymore. And we'll show this process in a little bit more detail when we get to that lab. Okay? So, great, I just wanted to take a second and kind of review enzymes and review proteins with y'all before we wrap up for the day, okay? So proteins, remember they are very complex and the most diverse of all the macromolecules. They have a lot of different jobs, okay? What makes up proteins? Well, they're made up of carbon, hydrogen, oxygen, and nitrogen. So the word I really want you to remember is CHON. What are proteins made up of? They're made up of CHON. Carbon, hydrogen, oxygen, and nitrogen, and sometimes sulfur. But for this class and the test you're taking at the end of the year, remember CHON, okay? What are the components of a protein? Well, we talked about this more last lesson, but the monomer of proteins are called amino acids. Amino acids come together. In between each amino acid is a peptide bond, and that is what proteins are made up of, are amino acids, okay? The ribosome is an organelle inside of the cell, and that's where we make our proteins. Okay, so this is very important about proteins. We kind of mentioned it earlier, that the shape of a protein is what allows it to do its job. So the shape of a protein is critical. If you change that shape, it's not going to work the way it's supposed to. And you can change the shape of a protein by high and low temperatures and high and low pHs. Okay, so we really want to keep your internal environment, like inside of your body, in those certain parameters. If you get too hot or too cold, these enzymes aren't going to work anymore and your body's not going to be able to function the way that it's supposed to. Same thing with the pH, okay? We don't want it too high or too low, okay? Lastly, we talked about something called the lock and key model of enzymes. This is when the shape changes, even if a slight change happens, the protein might not be able to work anymore. So just like your key unlocks one lock, it doesn't unlock every lock. That's very important to remember with these enzymes and proteins, is that the shape of the proteins are very, very important. Y'all, thank you so much for hanging in there with me today. Your teacher has some other work that I'd like you to give your best effort on so you can really understand this stuff. So make sure you give it your best, and I hope everybody has a great day.